Hello everybody, my name's David O'Reilly. I'm the Director of Scientific Research at BAT, and I'd like to start by thanking Euroscience for the kind invitation to allow me to speak to you today. This is my second Euroscience Open Forum conference, and it's always a great honour and privilege to present and take part in Europe's leading scientific forum. So today I'm going to talk to you about a novel plant-based approach to developing a vaccine for COVID-19. As of September 2020, there have been over 25 million recorded cases of COVID-19 around the world, and tragically, over 850,000 deaths. The world's scientific response has been stunning and extraordinary, and in the world of vaccines, there are currently around about 170 under development. And whilst that might sound a lot, it's a conventional wisdom that about 90% of vaccine candidates fail in development. So the world is going to need every shot on goal to make sure there's enough vaccine for the world's billions of people that need it. So what I'm going to take you through today are the advantages of plants for manufacture of biopharmaceuticals, and the tobacco plant in particular. I'm going to talk about the work we do at our US biotech division, Kentucky Bioprocessing. And then I'm going to take you through 3 case studies. The first is the Ebola case study of 2014, and the second 2 are our vaccine developments for seasonal flu and for COVID-19, and then I'll summarize. So why are we using tobacco plants to develop vaccines and other biologics? First of all, the tobacco is a model plant. It's well characterized and understood, and the genomes of several varieties of tobacco have been elucidated and published. Tobacco grows rapidly and can function as a bioreactor to produce high yields of biopharmaceuticals. It's highly competent at expressing, assembling, and modifying large and complex molecules with high fidelity. And of course, tobacco is not a food crop, so the acreage we use for this is not taking away precious acreage for food production. And importantly, there are no known human pathogens in tobacco that could contaminate any biological molecule or formulation. So Kentucky Bioprocessing is a company that's about 15 years old. We started working with KBP in 2009 and acquired the company outright in 2014. KBP is a global leader in cGMP production of plant express proteins. They have expertise in pharmaceutical-grade extraction and purification processes. And they're an innovative company, constantly looking at ways of creating faster, more efficient methods for developing, growing, and extracting biopharmaceuticals from tobacco plants. And over the years, they've built many deep and collaborative global partnerships, not just with the US government and scientific institutions, but also in Europe and elsewhere in the world. So let me now talk about the first case study for Ebola in 2014. So in 2014, there was an Ebola virus epidemic in West Africa, and at that time, KBP was working with ZMap Pharmaceuticals in California to develop a treatment for Ebola called ZMap. ZMap is a tripartite monoclonal antibody complex, and KBP took that, expressed it, and grew it and extracted it from tobacco plants. The US government allowed the use in emergency and clinical settings as a standard of care for treatment for patients infected with the Ebola virus, and many medical professionals and healthcare workers working in West Africa were successfully treated with ZMap. So that was the first case study for a plant-based biologic used in humans from KBP. Now let me move on to our vaccine developments for both influenza and SARS-CoV-2. Our vaccine system is based on what we call our TAP platform, which is the Tobacco Antigen Presentation Platform. The vaccine has 2 components, both of which are transiently expressed in tobacco plants, grown and then extracted. The first component is the tobacco mosaic virus scaffold. So that's expressed and grown in tobacco, purified and then inactivated. At the same time, in other tobacco plants, the antigen is expressed, grown and purified, and then chemically conjugated to the coat proteins of the TMV to create the TAP fusion vaccine. The entire process is very quick. From seeding the first plants to having the final product, it's about 6 weeks. And it only takes 1 week post-transfection for enough antigen or TMV to be grown in tobacco before extraction. So it's a very quick system. Now let me talk about our quadrivalent influenza vaccine development. We started discussing this with the US government in 2018, and they were interested in the potential benefits of the TAP system for treatment of seasonal flu. The main benefit that they saw was that because of the speed of which you could manufacture and extract from tobacco, that they could potentially select the antigens that would be used that flu season closer to the flu season rather than several months in advance. So with great support from the US government, we started our work in 2018. After successful preclinical testing, we entered clinical trials this year in June for phase 1. The preclinical testing with TAP-QIV showed favorable results. We saw promising immune responses that were stimulated with no toxicological findings. We saw prolonged immune response in animals, and the TAP-QIV vaccine appears to be stable at room temperature. Currently, we have 6-month stability data. We anticipate that could be as much as 12 months. So everything changed for us in January, as it did for many vaccine developers, with the publication on the 11th of January of the SARS-CoV-2 sequence. So KBP went to work with that sequence and created an antigen, manufactured it in tobacco, and by March we're in preclinical testing. In July, they submitted an IND to the FDA, and subsequent to that, in further discussions with the agency and with scientific bodies and regulators in Europe and elsewhere in the world, we decided to take a pause to modify our formulation and also modify our approach to clinical trials. And we're very grateful for that scientific advice. So currently we anticipate being in clinical testing during the autumn of this year. So let me talk a bit about the preclinical testing of our COVID-19 vaccine candidate. In preclinical testing, we demonstrated both immunogenicity and efficacy in multiple in vivo studies with no safety issues observed. We saw an extended residence in immune system tissues after administration, and we saw a breadth of immune response in vivo studies, both Th1 and Th2 associated antibodies. The immune response is stimulated upon a single dose with a strong boost response after a second dose. And the immune response recognized all sources of SARS-CoV-2 spike protein. And in our preclinical testing in vitro, we saw no antibody-dependent enhancement of disease. And as such, 3 manuscripts are currently under preparation, and we hope to publish those as soon as possible. So in summary, we believe the plant-based TAP system could potentially transform vaccine development and manufacture. It's an innovative plant-based bioreactor systems that have many potential advantages, such as stability at room temperature, safety because of no human pathogens, high fidelity because of the ability of tobacco to faithfully reproduce biologics, and other benefits. We think plant-based systems are highly suited to vaccine development and rapid response in situations where you have a new epidemic or pandemic. And of course, we think this is a potentially viable alternative use of tobaccos for vaccines, treatments, and other biopharmaceuticals. And finally, we think the TAP system is an innovative and pioneering vaccine platform with our first 2 vaccine candidates under development. Thank you very much for listening.